All right, here we go. I'm going to be able to speak today about some of the work we're doing at Recursion Pharmaceuticals. So my name is Mason Victors. I'm the Director of Data Science, Discovery, and Research there. And I'll be speaking about robust deep image embeddings, how we combine deep learning with a simple trick of a specific architecture called triplet networks to be able to mitigate certain experimental nuisance factors. So a bit about what we do as Recursion Pharmaceuticals. I'm going to talk to you a little bit about our problem that we're trying to solve and our proposed solution. Part of that solution involves deep learning and will heavily involve convolutional neural networks, as well as this concept of metric learning and triplet networks in particular. In addition to that, I'll talk about nuisance factors, explain what they are and how we can use triplet networks to help overcome them or mitigate those problems to a degree. And we'll also go through some of our results of our tests here. So Recursion Pharmaceuticals, what is the problem that we're ultimately trying to solve? Well, there are rare genetic diseases that affect millions of people. We oftentimes think of them as being rare because individually they might be rare. But because thousands of these rare genetic diseases exist and most of them are untreated, that means millions of people in the US alone are affected by these. Somewhere around 10% of people in the United States have one of these thousands of rare genetic diseases. And the traditional drug discovery model of one disease, one pathway, that's how Big Pharma generally approaches drug discovery. It would take hundreds of years to address all of these diseases. And we believe that we can do better than that. So our solution that we propose is this idea of disease models on demand through imaging. Why imaging? Well, it is actually an incredibly cheap way to get a massive amount of information about some condition. So you can think of it as we have a healthy child, and we can look at the cells from a healthy child, and they look like that top image up on the top right. But a child with one of these rare genetic diseases, in this example, one called Cornelia de Lange syndrome, their cells look fundamentally different. And that's because these diseases that we're modeling operate at the cellular level in that we're focused on monogenic loss of function diseases. So these diseases are ones where a mutation in a single gene causes that gene to be silenced effectively or lose its function. That means the protein that it encodes doesn't get created. And as a result, other bad things happen downstream. And so these downstream effects are visible in cells through our imaging technique. So we use something called cell painting, where we basically take different dyes that we can apply to a cell culture that we grow in our own wet lab at Recursion, and we can treat these cell cultures with these different dyes that will adhere or bind to different cell structures. So there's a dye that will fit in nicely in the double helix strand of the DNA. And another one that will attach to mitochondria or your actin fibers or your endoplasmic reticulum. And these stains are also set up such that if you excite them, then they will fluoresce at a certain wavelength. And so you can filter out all of the rest of the light, and you can basically just light up where the nucleus is, or light up where the mitochondria are, or where the endoplasmic reticulum is. And as a result, you can get these very rich images of a cell culture. So kind of the scale of our data, we think about one cell, and in an image there might be upwards of 1,000 cells in one of our images that we take. And we have about 300 different cell populations on a single plate, experimental plate that we run. And we might run anywhere between 100 and 200 plates a week. And what that does, it generates somewhere on the order of 125 to 250,000 images per week. And these images are about 2,000 pixels by 2,000 pixels. And so it generates a massive amount of image data in a very scalable manner to get this. And so in short, what we do is we take cells, we grow them and take pictures of them, extract information from those cells, and then identify what is the signature of each of these individual diseases that we're trying to model. And we look for drugs that when we add to the disease cells will make those cells look healthy again. And so in this case, it's right here how I'm depicting this kind of has two different parts. One, which is this feature extraction, pulling out the information from the images, and then the next step of it here, as described, is identify what the signature is from those features that we pull out. Well, that's kind of the old version of doing image-based analysis, of pulling out your features and having the features you pull out be completely static and hand-derived, human-engineered features. Well, that's kind of the original platform of recursion on the image analysis side of things. Is we'd have this original image and we would segment it. We basically use some classical image processing methods to identify where the nucleus is, and then identify where the rest of the cell boundary is. And once you've segmented out the nucleus and you've segmented out the rest of the cell boundary, you can ask hundreds of questions about each cell. What's the size of the nucleus? What's the shape of the cell? Is it nice and elongated, or is it pretty compact? What's the distribution of each of our 5 stains throughout the nucleus? Are they nicely distributed evenly throughout the nucleus, or do they congregate on the edge? And there are hundreds of these questions that we can ask, and so with the kind of fixed, static, human-engineered features, those are the kind of questions, that's the kind of information that we pull out from our images. Well, there's a problem with this. One, those human-engineered features are inherently static and cannot learn and improve over time. And the other problem is that segmentation in this way is quite fragile. If you're segmenting hundreds of cells in an image, then how you segment one cell might affect how the next cell is segmented and how the next cell is. So if there's small perturbations, it can actually create kind of an unstable effect in terms of how you extract information from the images. The other reason why segmentation can be problematic is it's not exactly necessary for what we're trying to accomplish. And there are many cell types that can't easily be segmented in this mode. And so it limits us to a degree in terms of the kind of cell types we can work with and what we're able to actually ask about these cell populations. So the solution, let's not segment. And that's where convolutional neural networks come into play, where we're able to learn the features that are most relevant to our tasks and not just rely on human-engineered features, and allow these features that are pulled out from the network to create this nice hierarchical and composited representation of our cell populations. And of course, we're familiar with kind of the standard building blocks of convolutional neural networks. And you put it all together, you create this nice layered hierarchical abstraction of of the information contained within an image. Well, deep learning has been incredibly successful in image tasks of a wide variety. Oftentimes, we think of ImageNet and that competition that occurs every year of this massive classification task. But deep learning doesn't necessarily have to be restricted to classification. We've already seen that many times in the past 2 days, where we've seen examples of people using it for segmentation as well, or for a number of other tasks. Another reason why we might not want to focus on just a classification task, even if you do have labeled data, is that there might be too many unique classes, or you might be constantly adding new classes to your data set, as is the case at Recursion. We don't have a standard set of 1,000 different diseases or 1,000 drugs that we have. Rather, it's on the order of tens of thousands, and it's constantly growing as we test new disease models and new drugs. So if you have an ever-expanding class or a number of classes, a standard classification task might not work very well. And I'll give another example later on around a deep learning approach that doesn't use classification as the main task for the deep learning problem. Another reason why you might not just want to have it be a strict classification problem is your classes. For each of your classes, you might have some external information that you want to model as well. So it's not just that you're wanting to predict what a class is, but you might want to take in other information to try to improve the way you represent your images in some kind of a latent space. So one approach of doing deep learning that removes this idea of a classification task is this idea of metric learning. Specifically with triplet networks is one great example. So triplet networks were this architecture that was proposed back in 2014, where rather than trying to determine what the class of some image was, you try to learn some way of representing distance between your embeddings or your representations of your network. So rather than just feeding in an image of a dog and asking, is this a dog or is it a cat, and telling your model to learn to say that it's a dog, what you would do is you would feed in triplets of examples. You would say feed in a base example, a similar example, and a different example. So examples of these would be a dog as your base, then a picture of another dog as your similar example, and then a picture of a cat as your different or negative example. And so you basically try to tell your network, I want my two pictures that are the same to look more similar to each other in this latent space than the picture of my base example and the negative example. I want those ones to look different from each other. So you're basically learning this kind of representation, and you can take your favorite distance, your distance function, your metric function, and you can learn some kind of representation there. So in your example, if cosine similarity is the desired way of representing your data, you can feed in that metric. If you want it to be Euclidean distance, you can feed in that instead. So what kind of loss function should you use here? So similar examples should have smaller distance than different examples by some margin. So it's not enough to just say, if I give in two examples that are the same, I want their distances to be close. Because in that case, you could learn a trivial map of everything to 0. And you'll have no error at that point if you're just saying that similar examples have to look similar. So you need to have some kind of contrastive piece here, which is where people typically use this hinge loss function, where you basically say that the distance between your base and your different example needs to be larger than the distance between your base and your similar example by some margin m. And so you can think of it as, before, if you have your base example at the center there on the left, and your similar example is that blue circle to the bottom right, and your different example is that red circle, you basically say that the distances aren't quite aligned, in that I need the distance between the two blue examples to be smaller than the distance between the blue and red example by some margin alpha there. And so you feed that in as your loss function to your network, and it then tries to learn a representation that follows this approach. So in fluorescent microscopy images, to talk about our use case in particular, we have our data on thousands of unique cell treatments across multiple cell types, and we have this problem that every week when we run the experiments, we do this kind of on a weekly cycle, there's always some nuisance factor, some experimental batch effect that tends to happen. I shouldn't say always, but it happens fairly often. Maybe the temperature in the lab is slightly off from where it normally was, or maybe there was some piece of equipment malfunction that made it so the time between step 1 and step 2 was an hour longer than it normally is. And you get these kind of batch effects, and that can be quite problematic. So here we have an example of cells, some treatment from week 1, and we look at a different treatment from that same week. And so this is a different disease model, and when you look at those, you probably can't see it very well due to just the projection on the screen, but you can't really see very many differences by eye. They're extremely subtle. The convolutional neural networks, they can pick those up. They can pick up the differences between them. But the problem is that if you look at the same treatment 1 on a different week, it might look fundamentally different than it did the first week because of some batch effect that might happen. And we call these nuisance factors. So we want to figure out how can we mitigate these or overcome these nuisance factors. And so with this problem of the same treatment yielding different phenotypes given different experimental conditions, it can prove somewhat of a barrier to progress in our drug discovery platform. So one of the most recent or the biggest, most well-known use cases of triplet networks was this idea of FaceNet that Google published a paper on this a couple of years ago, where they learn embeddings of faces for different tasks. And their reasoning behind this was that they have this incredible data set of millions of different images of faces of millions of different people. Well, you're not going to put a softmax layer trying to predict a million different people. That's going to totally make your model way over capacity to actually function well, and it might not even do what you actually want it to do. Your task might not be classification of who is this person. It might be some verification task or some clustering task or something else, where it would be better to have some kind of abstract representation of a face. And they show this example at the very beginning where they show, hey, our triplet network approach to this can overcome nuisance factors like lighting. They don't necessarily use that terminology, nuisance factors, but they give these examples of distances, of pairs of images and distances between those images. And you see that on the left we have all of these three people under one lighting condition, and on the right the same three people under a very different lighting condition and a different angle. And when you look at the distances, you see the distances between images of the same person, despite the lighting conditions, are smaller than the distances between people within the same lighting condition. So like, it's a pretty cool, cool result here. I thought, well, that's kind of interesting. In FaceNet, they use these triplet networks, and it helps overcome these nuisance factors, which are very similar to the kind of things that we have, these artifacts of the experiment. But in their case, their nuisance factors, they're present, but they're not really labeled. It's not like they get an image of a face and say, oh, it's an image from the left with the lighting coming directly on, and that's what I'm looking at, where they can take those labels to improve their way of overcoming the nuisance factors. In our case, they're present and labeled. We know exactly which week or which batch of experiments each image comes from. So we thought, well, can we take advantage of that? So let's suppose that xik be an image of treatment I from nuisance factor k. So say k represents our week of experiments or our experiment batch, and I is whatever disease model we're testing. So if we compose triplets of the form that I display up there, where you basically have your base be an example of some class from some given week, your similar is an example of that same class, but restricting it to be from a different week or a different experiment batch. And then restrict your different example to be a different treatment class, but from the same week or the same experiment batch as your original base image. And by doing that, the message that it kind of nudges or sends to the network is focus on inter-nuisance factor similarities and intra-nuisance factor differences, saying, I want you to learn what is similar between these two examples, even though I know that there are strong batch effects that are separating these. And I want you to focus on what is different between two images, even though they don't have that nuisance factor difference. And so you feed in these examples like this, and we hope that we find representations that are fundamentally better. So to kind of create an architecture for this and to be able to nudge it and help it learn faster and have some kind of intermediate feedback here, we basically created this representation model, which is some vision network that takes in an image and produces some representation. And from that representation model, we can feed in 3 different images at the same time. Your base example, your similar example, and your different example. And you get 3 different outputs. And then what we can do is from those outputs, we can calculate the distance between the base and the similar, the distance between your base and your different, and then calculate this triplet loss on there. We also added some very light, simple classification layer on top of that to help nudge the model forward a bit faster in its learning and to give some constant feedback to monitor. And what we found, and that faint line on the left are too faint to really see, but we found that turning on this triplet loss in addition to the classification task, it not only improved what we wanted it to improve of this cross-nuisance factor consistency, where we wanted examples of the same treatment across different experiment batches to look more similar to each other. It improved that. It also acted as somewhat of a regularizer for the rest of the network, where it was finding a better, cleaner representation. So it was preventing us from overfitting too much and helped us to learn better representations. Now, more recently since that work, we've continued to move forward with our deep learning platform and decided I'd give some kind of statistics of where we're at. So on the far left in red, that's an example of how, and I'm not giving this in terms of accuracy because our case, we don't really care about being able to predict what class something is because we know exactly what it is when we're treating these things. What we're wanting to know are how similar are two different examples or replicates of the same condition, and make sure that we're getting a very good, clean representation of whatever thing that we're actually modeling. So we measure this in terms of z-scores, of consistency relative to some null distribution. So on the left, we see that with Cell Profiler, which is this off-the-shelf image segmentation, cell segmentation based approach, we're able to get a pretty decent z-score compared to some more standard baselines, where you might just take like mean pixel intensity as a very naive approach. And that's what's shown in green. We decided, well, I wonder how good just randomly initializing some ResNet would actually do here. And it does about as good as just taking out the pixel intensity, which isn't that surprising. Well, then we decided, okay, where do we fall with our deep learning methods now? And we see that when we use a convolutional neural net to extract some underlying representation of the image, we significantly outperform Cell Profiler, and if we combine the two, we get an even stronger boost. So it's learning not just what Cell Profiler learns. Actually, it learns more than it, but it also misses some of the stuff that Cell Profiler gets. And combining the two, we actually get a much better representation of these cell populations that we're growing. I recently learned yesterday that one of my colleagues, James Taylor, just the past week outperformed these benchmarks as well. So this is already outdated with some of the continued work. And in my haste of preparing slides, I failed to acknowledge some of the work and much of the work that has gone into this, coming from the rest of my colleagues at Recursion Pharma. In particular, James Taylor and Brian Barg have been instrumental in developing our deep learning platform. So in review, I've talked a bit about Recursion Pharmaceuticals, what the problem is we're trying to solve and our proposed solution, as well as how deep learning plays into that, and this idea of nuisance factors and the results that we've achieved from that. And I'm happy to answer any questions. Also want to point out my colleague David Healy is here as well, so if I don't get a chance to talk or answer questions here, feel free to reach out to him as well.